Hello, everyone, and welcome to today's EAStalk webinar, part of our ongoing series by the European Aquaculture Society. My name is Anna Viskovic, and I'm the project and a program manager at the EAS. I will be your moderator today. EAStalk is a series of webinars that reflects a multidisciplinary nature of aquaculture and our diverse community. These sessions are designed to share knowledge, connect professionals, and provide meaningful insights for students, early career researchers, and experts across the sector. Today's session will run for 45 minutes, including a 30 minutes presentation, followed by 15 minutes live Q&A. During the Q&A, you're invited to share your questions, so please share them in the chat below. Please note that this session is being recorded, and the recording will be available on the EAS YouTube channel for 1 month, after which it will be moved to the EAStalk webinar section on our website, which is accessible to the EAS members only. Now, let's turn to today's exciting topic, anti-pathogenic mechanism of nature feed products in fish and shrimp aquaculture. With the growing concerns about antibiotic resistance in aquaculture, there is a pressing need to find effective alternatives. Today's webinar will focus on natural antimicrobial feed additives, such as essential oils, organic acids, and plant-based extracts, and how they can reduce the virulence of harmful microorganisms while improving gut health and animal performance. We are thrilled to welcome today's presenter, Professor Nicolae Corcionivoschi. Professor Corcionivoschi is the head of the food microbiology at the Agri-Food and Biosciences Institute in Belfast, Northern Ireland. He is also a fellow of the UK Higher Education Academy and an elected member of the Royal Society of Biology. His impressive research spans bacterial virulence, pre- and probiotics, foodborne pathogens, and gut microbiota, all within a strong framework of biotechnology, molecular biology, and pharmacognosy. He's also deeply involved in training the next generation of researchers through his supervision and teaching roles in several universities across Europe. Today's webinar is presented in collaboration with Auranta, a corporate member of EAS, and a part of their membership benefits is this presentation. You can learn more about them by visiting their website, which I will be linked to the chat. Professor, the floor is yours.
Thank you very much, Anna, and thank you for inviting me today. It's a pleasure for me to present today the work that we have done over the last 10 years in this area, especially most of the results you will see today will refer to work done by PhD students and postdoctoral scientists. So, so at AFB, my research group is trying to cover 5 big strategic transitional research groups. We're looking, for example, at the impact of antimicrobials on gut health, trying to find effective management practices, trying to understand the host microenvironment, ultimately trying to design, as I like to call them, intelligent interventions. And we do all this mainly to try to reduce pathogen and their virulence. We have designed and we have in vitro, ex vivo and in vivo models that helps us understand the biological mechanisms behind the mode of action, and ultimately trying to design these novel interventions. And some of the areas we cover is essential probiotics, antimicrobial molecules, or mixtures of natural antimicrobials, like plant extracts. So why we do that? Well, one of the reasons we do that is because we know that antibiotics are a real challenge to farmers nowadays. You know, there's an immense pressure to reduce the level of antibiotics that we use in farmed animals. So what we try to do is to look at natural alternatives. And for example, you see here in this slide, this diagram of this biological mechanism, which actually shows you how, for example, natural microbials work. And here you have essential oils. And if you try to remember yourselves how antibiotics work, you will see in this image here that actually natural antimicrobials, they in a way act against pathogens in a very similar way. Why we're trying to do that is because by introducing these novel alternatives, or these alternatives in combination with antibiotics, you might be able to actually reduce the level of antibiotics and reduce the spread of antibiotic-resistant genes. Because in 1945, for example, Fleming was giving an interview to the BBC saying this. So finding the right dose, that is a crucial.
I was not able to hear the sound. I'm not sure if maybe
it's okay. Basically, it's okay. Basically, what Fleming was saying in 1945. Basically, what Fleming was saying in 1945 at the BBC was he was saying that if the dose is too small, the microbes will not be cured, and there is a risk they can gain resistance to those antibiotics. So basically, that tells us as scientists nowadays that finding the right dose, understanding the mechanism of action of this, especially of this novel alternative, it's crucial. So there were three questions that we tried to answer. We're doing a bit of brainstorming, trying to answer three main questions in our research. Why do we actually need these alternatives? Well, we do. How do they work? And are we actually using the necessary approaches? And are we actually using that? So basically, we're in collaboration with many industry partners. We're trying to focus on different farmed animals, but today I will talk about the work that we do in aquaculture. Why we do that and why we think at the institute level is important is because over 50% of seafood consumed, for example, globally, now comes from aquaculture. So it's definitely an important sector. And you could see here in this slide the market value as recently published. And it matters to us because we know that we're trying to create climate resilient spaces. They support rural economies and trying to reduce antibiotic reliance. And by doing so, we are enhancing environment and trade benefits. So aquaculture is a huge sector. And the work that we're trying to do in this area, we believe that it is very important. So in part one, I will try to present to you some of the work that we've done using antimicrobials in shrimp aquaculture, basically trying to understand through models of infection or ex vivo models, trying to understand the biological mechanism that sits behind the mechanism of action. And in shrimp, and trying to eliminate the cause of infection. So we looked at parasites and microsporidiums. For example, some of the results you see on hematopsis, on EHP, both important diseases in aquaculture. And also we're looking at viruses and bacteria. Some of the work that we've done with collaborators, looking at the white spot syndrome virus and the new vibrio parahemolyticus infections. So you have seen this slide here, why we're working on this, very important, and I'm sure because you all work in this area, you know the importance and the devastating effect that these infections might have. So natural mechanisms, we mainly work in mixtures. Why in mixtures? Because all the literature nowadays is telling us that they work better in that way. The mechanism and the efficiency is much more enhanced when you work with them in mixture. So once you see an example of the mixtures that we can be testing and we have tested and published, including citric acid, sodium citrate, malic acid, citrus extract, olive extract, or maltodextrin or silica, and the so-called product, our aqua, and more information can be offered to you by our roundup. the advantage of using this type of mixture is that it's non-antibiotic, it's plant and organic acid based, there's no residue risk. So this is the result. If we going back to, as I said, if we're looking at the mitosis, for example, the mechanism of actions that we have identified, it blocks parasites addition to epithelial cells and strengthens the tight junctions, the transepithelial resistance, as we call it. And you could see it here in this figure here in this diagram, that when a parasite, usually when it infects cells, it polymerizes the actin, the so-called actin polymerization. The moment it does that, disrupts these tight junctions, and you could see here the impact that they have in blocking some of the proteins in the tight junctions, and by doing so, they cause infections and they trigger a chain of, let's say, explosive results within the cells, where they trigger this host cell ADPH oxidases to produce nitrogen peroxide, and then through this externally regulated kinases, they're actually able to produce this, stimulate the host to produce these antimicrobial peptides. What we showed and what we have published with products like our Aqua, for example, we show that the moment you do that, you block this actin polymerization, and some of the enzymes, like you modify, for example, the activity of the excess of the ERK, of the externally regulated kinase of this ERK enzyme by dephosphorylating it, for example, and you see no need for these antimicrobial peptides to be produced. So basically, you see this increment in antioxidative activity within the cell, and as far as we know, this was translated then in vivo as well. So I have already described some of the in vitro key findings. However, when the studies have done in vivo, we could see that in terms of mortality, in the presence of this mixture of natural antimicrobial, we go from 68% to approximately 14%. And also in those in this infected shrimp, we've noticed as well an increase in antioxidative activity. More so in these trials, we've seen a change. The scientists, the students have also looked at the impact on relative growth rate, on daily weight gain, on total weight gain, and feed conversion rates. And I will only talk about FCR, feed conversion rate, and you could see that there's significant reduction in FCR, and then an increase in survival, and also in terms of production significance. Also, you could see that in terms of length of the shrimp was also improved. So there was significant result that could have been, and they were translated from biological mechanism, had a biological mechanistic relevance in vivo as well. In terms of gut health and gut microbiota impact of this product. So, as you know, giving probiotics to an aquatic creature is not an easy task, because they have to go to feed, and the moment they reach water, some of these feed pellets might not all reach the host. Or anyway, it's a difficult thing to apply in aquaculture. However, we took a different approach, and we said, how about if we find ways to stimulate the host, the self-born probiotic microorganisms that can actually impact, for example, impact on infections like the vitro parahemolyticus infections. And we have noticed in the literature is well known that Helicobacter pneumoniae, for example, is a probiotic microorganism that sits in the gut of the shrimp. However, when it's stimulated, it's actually able to grow and develop within the gut. So the question that we had, can we actually overstimulate that production? And in vitro, we show that indeed natural antimicrobials are able at the various concentrations to improve the growth of this probiotic when it's in single culture. And so you could see it translated here in the colony forming unit. So at 24 hours, there's a significant increase in growth of this probiotic microorganism. So we have designed a very fancy experiment, for example, because obviously they need a substrate to grow. And the most important thing is this F. prausnitzii, this probiotic microorganism produce butyric acid, which we know it's a very important immunostimulant. So we have designed this very fancy experiment here, where we actually show that this, in the presence of our aqua, in the presence of this mixture of natural antimicrobials, regardless of what they are, actually, you know, you increase the activity of this probiotic microorganism and an increase in substrate usage. And also this was translated in an increase in butyric acid production. So you could see an increase in usage of the substrate and an increase in butyric acid formation. So this was actually a very promising result to us. And then we moved to a different model that we have used. And here we looked at, is this translated then in an inhibition of infection or a reduction in infection? And indeed, I won't go through all this figure here because it's quite complicated. However, what we did here was to see, for example, if we have pre-treated a probiotic with sorry, apologies, with natural antimicrobials, and then we colonize gut primary epithelial cells with them, and then we infect with Vibrio parahemolyticals. What we noticed is that we reduce both addition to the cells, the ability of Vibrio parahemolyticals to adhere to these cells, and also to internalize. So that's how many cells are on the top of the cells, or how many cells are actually internalized. And what we've noticed when we do that, then we have this increase in butyric acid formation. So this was again a very positive result and published in the Journal of Biology in the last couple of years. So in terms of the mechanistic insight, it was clear that this type of antimicrobial mixtures are able to stimulate, for example, host-derived probiotic microorganisms, and then to have a detrimental effect on addition and invasion. And we have, in that way, we have offered and we opened the gate for a different type of intervention at a different type of intervention at farm level. If we're moving then to EHP, another significant and important in shrimp aquaculture, we know that I'm only going to summarize the mechanism here for time's sake, to try to save some time. However, we have noticed through our in vitro and in vivo experiments that indeed we can reduce this devastating impact of EHP. And we have also basically the mechanism is described in this figure here. But basically, when you have antimicrobials present in an infection assay, you block this actin polymerization. So the actual organism is not able to attach to this cell, and then is not able to trigger this oxidative stress mechanism within the cell. And the way it does that, in the case of EHP, interestingly enough, is by blocking the NF-kappa B complex, and that is important. Why? Because this is the system that is important in shrimp, for example, for activation of antimicrobial peptides. So this reduction in oxidative stress and then the less need for antimicrobial peptides to be produced was also observed in vitro. So indeed, this important effects and this clear effect were also published as well by my students, and actually were also significantly for the industry against their efficacy in vivo as well. We're moving a little bit now, and I'm going to move a little bit further, talking about viruses a little bit. I know we're moving now aside from the issue of antibiotics, we're talking about viruses. However, we know that sometimes, bacterial infections and viral infections, they do work concomitantly. So there's also one infection comes on the back of the others. However, in the efficiency, and I think the results that we have on white cell syndrome, it's very clearly described here in this figure. Basically, if you have an uninfected, organism, what you'll notice that we have this nice and thick mucus layer on top of the cells, no oxidative stress, and a low expression of interleukin IL-22, which is a marker for white cell syndrome infection. So in shrimp that have this type of infection, usually you have high levels of IL-22. So we have used that as a biomarker protein in studies. However, when infection occurs, you could see that the mucus layer gets really thin. You could see that IL-22 expression as the coagulation is going up. You see a lot of hydrogen peroxide produced and higher activity of the superoxidase and catalase enzymes, to reduce the detrimental effect of hydrogen peroxide, obviously. However, what we notice that when we have an infected organism and we have it in the presence of these mixtures of natural antimicrobials, what we noticed through our experiments was that we recover this mucus layer. And not only that, but because we recover that mucus layer, the virus is less able to infect these cells. And also, there's a significant reduction in oxidative stress and IL-22. So that was a huge, that was a massive result for us. And again, successfully published, peer-reviewed, and all these papers are available for you to read in further details. In that, from that point of view, then this result was also translated in vivo, and in an in-house experiment when we had an uninfected control, an infected control, and an infected experiment in the presence of natural antimicrobials, we were actually able to reduce in a dose-dependent manner the impact on mortality, which was quite significant, from over 96% to 7.3% mortality. And these results were also translated in on-farm trials. So significant results. Again, they're all published in Antioxidants, so they're all there for you to read in more in more details, more detail if necessary. So then we're moving. Recently, I'm not sure how many of you are aware, but recently a new strain of Vibrio parahemolyticus is having devastating effects in Far East Asia. Basically, this strain is actually able to cause this, to disrupt, to cause this so-called translucent post-larval disease, when shrimp are becoming translucent. I don't have a picture here, but I'm very sure that most of you have heard of this. So we have taken that approach to actually see if these natural antimicrobials have and can potentially have an impact on this particular strain, And what we have seen is that indeed we are actually able with natural antimicrobial to even kill this pathogen. But also when we're looking at infection, in infection studies, we can see that we can reduce the adhesion of this pathogen to primary epithelial cells and also the ability to invade cells. And this also resulted in a less decreased levels of oxidative stress. And if we looked a little bit further into stresses, these are some of the genes that have been described in a few papers as some of the virulence genes, very important in the pathogenicity of these microorganisms. And we have actually, again, able to, as you can see here, to reduce the expression significantly reduce the expression of these genes concomitantly with at 45 hours post-challenge, for example, to reduce the oxidative stress in this shape. So that gave us a huge confidence. And again, these results were published. We also looked at mortality as well. You can see dose-dependent mortality, again, with the highest impact at about 1%. Antimicrobials. I'll move a little further because I think I'm getting short of time, Anna, if I'm not wrong. But
7 more minutes.
Yeah, so I will try to then move and try to show you some of the results. Some of the results we have in fish as well. But this slide actually only summarizes a little bit that's what I have showed you and the impact that this mixture of natural and antimicrobials can have by knowing very well the molecular mechanism that sits behind the mode of action. So in the last part of this talk, I want to talk about their impact on fish aquaculture. Again, I won't go to this introduction in detail, but what we looked at Priscetia salmonis and Streptococcus galactiae challenges, and again, very important infections, especially in salmon and tilapia, and also we look at Lactococcus garvia, again with devastating impacts on fish aquaculture because they're responsible for causing this hemorrhagic septicemia. And in terms of Priscetia, again, this is the first, we can see that if we look at the pathogen itself, we have detrimental effect on this ability to grow. We have a minimum inhibitory concentration of approximately 1% and a killing concentration about 2%. However, the biological mechanism of action in this case using fish cells engages basically weaponizing the host nitric oxide. So the natural antimicrobials are actually able to trigger this nitric oxide, which then has an impact on lipid peroxidation and bacterial killing. So this is, in very few words, the mechanism of action. Again, this work has been published and is available for you to read in details, but for sure it has an immunomodulation and an anti-inflammatory effect, decreased expression of inflammatory cytokines, as you can see, interferon gamma, IL-8, IL-2, IL-12, and IL-1 beta, they go down in the presence of antimicrobials, and we get this increase in the nitric oxide formation. So that is weaponizing that system to reduce the impact of disease. In Streptococcus galactiae, again, these are just impacts. This slide here only shows the impact of natural antimicrobials on the pathogen itself. And again, you can see that in infection studies, in vitro infection studies, you could see there's a significant effect at various concentrations, and there is no impact on host itself, so the host is very much protected. However, why we believe there's a mechanism, a game changer as we call it, because in this case, it's actually able to reduce the capsule polysaccharide formation. So this is a capsule on the surface of the cell, which is disrupted, a major virulence factor, and which then results in reduced inflammation. So we're actually able to reduce in the presence of these natural antimicrobials to reduce the formation of these pro-inflammatory cytokines, and you can see here the reduction in the impact on the oxidative stress as well. These are very fast experiments. Again, I won't go in much detail, but we have now a model, a tilapia gutfish model that we can study, and we can see here. I only focus on this part here on A2, and you can see that in the presence of natural antimicrobials, if you have bacteria that you treat with natural antimicrobial, you reduce its ability to infect and to this gut tissue, reduce the inflammation and reduce the oxidation. And these results are actually also seen when the antimicrobials, the natural antimicrobials, are added directly to the infected gut. So in a way, suggests that these mixtures can easily be used as prophylactic treatment to prevent infections. Again, if we look at disease, we look at two different markers, MCP8 and DUO1. These are biomarkers for disease in tilapia. Excuse me. And again, we could see that both of these markers are significantly reduced in both cases when you add the antimicrobial mixtures directly on the treat the bacteria, the pathogens. So obviously, this we have published this in English Material Journal recently as well. Less oxidative stress. So basically here what we use the gut model and the tilapia gut primary cell model. And basically what we show here, indeed, that we reduce in both models, in the presence of these antimicrobial mixtures, the oxidative stress was significantly reduced. In infection with Lactococcus gargia, again, I will move straight to this slide here because I think this is the most relevant. But actually what we show here is the prevention of red blood cell lysis. So basically, because it's a hemolytic pathogen, we have designed an experiment and we clearly show that when fish red blood cells are in the presence of the pathogen, you could see that the lysis happens. However, when you add the natural antimicrobial mixtures, that lysis, that disruption of integrity of those red blood cells is prevented. And you could see that you could see why, because when we looked at points of genes, they are all significantly downregulated. And again, this work has been peer-reviewed and published and available for you to read in more detail. And this figure here describes what I actually just presented to you. This protective effect that the natural antimicrobials have when present in a hemolysis experiment. So I'll just try to summarize that to say that to practical applications, obviously these natural antimicrobials they can be treated as feed additives and they can be used as prophylactics during the early stages of infections. They indeed are becoming more and more a preventative tool in high-risk aquaculture zones. And indeed, mucosal immunity enhancers, they can become mucosal immunity enhancers when we pair them with vaccines. And we have done some work in this area, but that presented here. So in terms of industry relevance, again, we believe that this will become more and more relevant. And then especially because they reduce mortality at such higher level. And I believe that now, because we understand the mechanisms of action, the practical application becomes easier because we have that strategic mindset. For example, the work that we've done here was don't kill the microbe, disarm the pathogen, disarm the invader. So in a way, we don't work as an antibiotic does. An antibiotic kills the pathogen. Well, the approaches that we looked at is to disarm these invaders, the so-called pathogen. And again, why does it matter? Because it reduces mortality, no withdrawal period, maintains economic flow, and again, increases consumer trust as well because of their ability to reduce antibiotic treatment. And these are just some of the papers that we published. In the background, you just see a microscopic picture of what the pathogen does to an epithelial cells when it infects, and we described a lot the antioxidative stress here. You could see a pathogen here invading an epithelial cell. We could see in green, in bright green, this host endothelial oxidases being triggered and basically being the ones responsible for reactive oxygen species release. So in a way, all the work that you have seen today fits into that area of One Health, and we believe that we will gain more and more experience in more and more visibility in the next few years. I would like to finish by thanking the founder. Orada has sponsored this work to many PhD students and postdoctoral scientists. Some of them are now doctors, as you could see here, the 3 students that graduated in the last few years and have been responsible for some of this work. So thank you, everybody, and I'm happy to take any questions now.
Thank you so much, Professor Corcionivoschi, for the fascinating and insightful presentation. You have clearly shown how natural feed additives can play a critical role in reducing pathogenically, improving gut health, and potentially offering alternatives to antibiotics, which is important in today's aquaculture landscape. I would like to open the floor to the questions, so please feel free to post your question in the chat. To break the ice, I would like to ask you a first question, and that is, can, in combination, natural antibiotics increase the efficiency of an antibiotic?
Yes, this is an important question that we try to find an answers for years. We do have evidence, unpublished evidence at the moment that, for example, just to pick an organism, for example, against listeria and it working through pathogen, that if we do use antibiotics in combination with natural antimicrobials, we actually can reduce the level of antibiotic use significantly, resulting in pathogen killing. Yeah. So they can be used in combination, and will offer the possibility to reduce antibiotic usage.
Thank you, Professor. I would like to encourage everyone from the audience, if you have any questions for Professor, please write it down. Now is the time to tackle all the questions that you might be having. So please start typing the questions in the Q&A. My second question would be, how do natural antimicrobials perform against pathogens in other farm animals?
Yeah, as mentioned at the beginning, we have done work in large farm animals, like we have done work in dairy, in cattle, and we have done work in poultry as well. And for example, in poultry, we had really significant results with Philip, one of the students in the slide. He has actually looked at, for example, the impact on Campylobacter levels, or we're looking at highly virulent Campylobacters in poultry and important pathogens, as you know, in humans. And
Professor Corcionivoschi, I only have one comment. Can you please put your microphone a little bit?
Can you hear me now?
Yes, I can.
Can you hear me better? Yeah.
Thank you.
So yes, they do have an impact. So we've seen that in against the Campylobacters in poultry, and also we've seen a significant effect against Cryptosporidium infections in cows. So yes, they have work has been done in that area as well. But Alexander was one of the past postdocs that we had funded as well in the past. Yeah.
Thank you. While we are waiting for some questions to receive, I mean, this can be, or that it seems that presentation was quite clear, and participants do not have any questions for you, but still, okay, there is one We just received one. So the question from Simona Sushnik-Baetz, have you perhaps studied the effect of the mixture of organic acid on any other biological processes, for example, reproduction?
We haven't specifically looked at reproduction. This is something that we have discussed, and we have that in plans, for example, to look at layers, for example, like in chickens producing eggs, to put it that way, there's some infection in there that in the parent flock, for example, there's a disease, there's a Campylobacter hepaticus, for example, is responsible for a reduction in number of eggs laid, let's say, in this flocks. And we recently have unpublished data at the moment that we are trying to complete, but basically we do have evidence to say allows us in a way to say that it has an impact on Campylobacter hepaticus as well. So by extrapolating, we potentially believe that on the reproduction system, it might have an impact. And I mean, and in a way, let's be honest, I mean, if the welfare of these animals is improved, you will expect that that reproductive system will also benefit from all that boosting in health and welfare.
Thank you, Professor. The next question we got is from Angela. Regarding the product's effect on mucin production, does it also induce mucus production in the gill, specifically for tilapia?
We haven't looked in the gills. It's a good question, but what we know from the gut is all the mucin genes are upregulated. So there is an expectation that some of these genes are also responsible for the mucus production in gills. So I wouldn't see why. We haven't measured in the gills, but because we have that massive increase in gene expression in these mucins, you would expect to see that as well in the gills. It's just the logic extrapolation that I'm making, looking at gene expression levels.
Thank you. The next question we got is from Liviet Dunk. Have you tried with EHP or just mechanism? I do not see your experiment with HP.
Sorry, Anna, can you repeat that question? Is it written somewhere, is it?
Yes, you can find it in the Q&A. If you click on the Q&A. It's from Leviet Dunk.
No, I don't see the question, so maybe your posts. So yeah, I said, have you tried? Yes, we have done with HP, and the presentation contains information about. We've done with PHP. Maybe we haven't included all the data in there, but again, like if you email me directly, we can provide you with more information on the HP.
Professor, if you don't mind, can you please put the last slide of the presentation back? Because there is your email. So
I don't know if you can see it.
Yes, we can see it now. So in case you would like to email Professor later on, feel free to mark his email.
Yeah, and we can provide you with all the information.
Thank you. Can I start with another question?
Yeah.
Perfect. So that's from Marco Sargolia. Thanks for your interesting presentation. I have two questions. So the first one is, do you have any idea about the cost of the AA application in feed?
I don't, but again, As far again, if you email me, I can pass on your question to Rwanda, and they will have all this information for you. Because they have been in charge in running all the farm trials, so they have all this cost, so they can provide you with information regarding the costs for the application.
Thank you. So the second question from Marco is, you say that it does not affect the microbiota, but just stop the invader. I cannot understand how the microbiota may not result affected.
It's possibly, you know, like, because we know as with prebiotics and as with different supplements, pathogens in general, they are not residents in the gut. Most of them are recolonizing the gut. The host itself, wherever the microbiome establishes, it also creates a protective system against, and then also healthy microorganisms. They have different biological mechanisms of actions. However, we're talking about opportunistic pathogens. Pathogens are not usually there. They might be, and we have seen beneficial. Again, there's a paper that my group has published in the journal. You can look for 2 papers in the Journal of Microbiome, a highly respected journal, and you can see that we have published, for example, recently the impact of different types of interventions on the poultry, for example, microbiome, and that could explain to you better, in a way, why the microbiome itself is not affected in a detrimental way. I guess your question goes into that direction. You would expect to see a detrimental effect on the gut microbiome. Yes, you do. However, not at the extent that could impact on the health of the host. And you could see, but that microbiome paper actually will explain to you much better what I mean by this opportunistic pathogen being treated in a way, in a different way in that niche. I'm not sure if I'm answering the question very well, but again, if I refer you to these 2 papers, you will understand better what I mean by that. And it is a great question, you know, it's on everybody's mind. But we have seen with other hosts as well, that the microbiome is not impacted in a detrimental way.
Thank you so much, Professor. The next question we got is from Farhana Ahmed. Can these natural feed additives have adverse effects on beneficial gut microbes? Sorry.
It's basically the same question as the previous one. Again, it might have on 1 or 2. Even if you do microbiome studies, it's very difficult to pick up the impact on everything. However, again, to our studies and to many other studies published, you know, the detrimental effects have not really been seen or described sufficiently to understand that.
Thank you. The next question we got is from Leon Krijman. Is it possible for pathogenic microbes to develop a resistance to the suggested antimicrobials?
Absolutely, fair question, and I get this question a lot. If you remember what I said at the last part of my talk, I said that we don't use concentrations to kill the pathogen. We use concentrations to design the pathogen. We use concentrations in which we're trying to play hard with the biological mechanisms present in these pathogens. That's why we're trying to design interventions, as I like to call them, to design smart and informative interventions in which you don't play with the impact, with let's say the antibiotic effect of that you see when you use an antibiotic, right? So we use concentrations, sub-inhibitory concentrations, in which we play with the biology of these microorganisms, so which is giving you less likely resistant possibility of resistance in the future, which goes back to, again, to Fleming's interview back in 1945, where he actually, in a way, says the same thing. That I'm comparing myself to him, but I'm just putting it in way, you know.
Thank you. The next question we got is from Rasadian Gassim. If you could please explain more how nuclear factor kappa B pathways is blocked. Is it a positive effect?
Well, it is a positive because NF-kappa B, it's a system that is activated by hydrogen peroxide, by reactive oxygen species. So when an infection occurs in the cell, these infections trigger hydrogen peroxide, triggers this phosphorylated pH oxidase to produce hydrogen peroxide. So it's a reaction to stress. However, these reactive oxygen species, they then activate this NF-kappa B system. So the NF-kappa B system then triggers a pro-inflammatory effect. So all these pro-inflammatory cytokines will be expressed. What we noticed with this natural antimicrobial, because we block the infection, because we block the ability and to reduce hydrogen peroxide, obviously by having less hydrogen peroxide, then this NF-kappa B system is automatically activated, even if the pathogen is present, but is less able to induce this pro-inflammatory effect. That's the very quick answer to that question.
Yeah. Thank you. The next question we got is from Lauren Chapman. Have you checked the effect on acute hepatonephrotic necrosis disease?
Yes.
Toxin production.
Yeah, the straight question is yes. I've said that in the presentation, somewhere in the presentation, that indeed that toxin production is decreased. Yes, so yeah, the straight answer is yes.
Thank you. The next question we got is from Nain Fana. When is the suitable time to administer the natural products, before or after the infection?
As mentioned during the presentation, we have seen better results in every model that we've used when, in both cases, when we pre-treat the pathogen or with the antimicrobial, when the antimicrobials are present during infection. So we believe that the prophylactic way of using this is more beneficial, because you tackle a pathogen. Imagine that you have a cell and you have a pathogen coming in to infect that cell. Obviously, that pathogen goes in the cell and starts multiplying itself. But if you have that, if you make that pathogen unable to multiply, to produce toxin from the very beginning when it's a low number, for sure your impact is much more significant. So yes, indeed, the prophylactic effect, I would strongly recommend, yeah, prophylactic way or prophylactic usage of the
Thank you so much. I will read now the last question for today. So in case you might be having any other questions for Professor, feel free to mark his email and email him after this presentation. So the last question for today is from Livet Dunk. So there are actually two questions. Basically, the product contains organic acid. So could it be classified as a natural? Farmers will need to clarify the natural versus synthetic one.
Yes, because they're plant extracts. You can have them. You can have these acids either synthetic or organic. Usually, we use once you use organic extract, with containing this organic acid, also they will be classified as organic. Depend again. You have to check where you buy them, from which form they are. So yeah. Or, obviously, it's far-fetched, but, there are papers there suggesting that you can nowadays create microorganisms to produce this type of acids.
Thank you. And the second part of the question was, we use lots of organic acid mixture products such as from Elanco company in Vietnam, but cannot treat or prevent HP.
I can't really offer an answer on that because I don't know exactly what are they, how they use, so I can't really comment on that. Question is very brief, so I need to understand more about whatever it's used,
Thank you. Thank you for joining us today, and a big thank you to Professor Nicolae Corcionivoschi for sharing the expertise and taking the time to answer all the questions that we received at the end. I think we have learned a lot today about the interdisciplinary strategies behind the natural antimicrobial feed additives. I would like to special thanks to Auranta, a corporate member of the EAS, for supporting today's webinar and for contributing to knowledge exchange within our community. As a reminder, this webinar is recorded, and I will send you the link of the webinar recording via email. And I hope to see you soon again in one of our upcoming EAStalk webinars, and of course at Aquaculture Europe 2025 in Valencia, that is happening next week. Until next time, thank you and take care and have a lovely day.
Thank you.
Thank you.